Hello, everyone, and welcome to the National Association of Plant Breeders webinar series, How to Breed New Plant Varieties, Imagining and Engineering Crops. My name is Kim Sher-Latier, and I'll be serving as your host today. This is a special presentation presented by Neil Anderson of the University of Minnesota. He will be speaking about ornamental flower breeding. The title of his presentation is From Lilies to Gladiolus, Flower Power. After the 30-minute presentation, you will have the chance to ask questions. We'll answer as many questions as we have time for in the question period. So now I'm going to switch control of the screen over to our speaker.
Pleasure to join all of you today to talk about the fun things we do in flower breeding and genetics and breeding ornamental crops for the market. So if you see on the first screen just some examples of the products that we have been developing and are developing in the University of Minnesota Flower Breeding and Genetics program, which has been around actively since 1926. In the lower right-hand corner, you'll see some chrysanthemums. These are of our new series of mammoth garden chrysanthemums that are winter hardy and form shrubs in the landscape. We've been breeding chrysanthemums here at the university since 1926 and are well known for winter hardy types. But today I'm going to focus on some other crops that we've been breeding for the past 15 years at the university and some new product types that we think are of great interest to the market. And that includes gladiolus, an example and a wild-looking flower of a gladiolus is in the top right-hand corner. And then just to the left and down from that are some examples of seed-propagated lilies, which we'll also be talking about today. The horticultural market, particularly flowers and ornamentals, are driven by novelty. And this chart just reflects some of the research that we did a few years ago about the number of products that are on the market in North America. And this chart just shows that since 1987, where we had approximately 20,000 cultivars and products within horticulture on the market, that this has grown significantly over time, such that by 2008, we had approximately 105,000 cultivars or species of horticultural products on the market. And this includes both flowers and ornamentals, as well as fruits and vegetables, landscape grasses, and trees and shrubs. But a large bulk of the horticultural products that are on the market are ornamental flowering crops that we call floriculture. Every year, growers have to pick and choose between thousands and thousands of cultivars as to what they're going to grow and sell and bring to the market. So we're just going to talk a bit about some of the processes that we do, what's been some of our keys to success, and where we're headed in the future. Some of the keys to our success come from our excellent tradition of breeding and genetics in floriculture. For instance, F1 hybrids, which are the result of crossing inbred parents that are unrelated together and creating a new hybrid, which has hybrid vigor, and that is far better in performance than either of its parents. And the first F1 hybrids in the flower world came onto the market in the mid-1950s, and that was the invention of F1 hybrid petunias and impatiens. And those were done by Claude Hope and Charlie Weddle of Pan American Seed Company in Costa Rica. And all of the products that are on the market today in the floriculture world are predominantly F1 hybrids that may be seed propagated or they may be vegetatively propagated, as you see online too. Other things that have created vast interest in ornamental products, creating these thousands of cultivars that are on the market, are the plug revolution, the invention of plastic and creating it such that we could use containers and plug trays to produce plants, has allowed for massive production of plants in greenhouses anywhere in the world that can be shipped easily um, offshore or within countries. Automation has certainly helped us in that realm as well, where we can automate almost everything in the process, from sowing a seed and germinating it to producing plugs that can be sold and grown by growers, to finished products, and then the shipping of the finished products to the consumer. The use of soilless media versus field soil that has been pasteurized has created lighter mixes, meaning that we could ship material farther and farther distance-wise because of lighter weights. Enhanced post-harvest quality has allowed for flower products to last well when shipped across the country or across the globe, which has created a global market. And then, of course, innovations in shipping have certainly helped as well. Next. Probably the inspiration for most of the flower breeders in the 20th and 21st century hail back to the practices of Luther Burbank, who was a flower breeder from Massachusetts who settled in Santa Rosa, California, and until his death in 1926 produced approximately 1,000 cultivars of every genus of flowers from A to Z, from Abutilon to Zinnia. And his practices and many of his methods that were used are still used by flower breeders today in both the public and private sector. And so these are just some historic shots from his flower breeding on the left. That's him breeding some calendula, as well as some other historic photos from his production facility. Next. To give you an idea of what flower breeders work with when it comes to producing new cultivars and for the ornamental market, regardless of what it is that they're breeding, and it might be bedding plants, it could be herbaceous perennials, it could be cut flowers, it could be foliage plants, it could be flowering potted plants. Regardless, we usually start with what sort of category it would fit into as a new cultivar. So if you look at the lower left-hand side of this slide, typically what we produce is what's known as a series. And that's a set of cultivars that match each other for most traits, with the exception usually of one, differing oftentimes by flower color, creates a series. And every series will have a name. So like we might have, for instance, a chrysanthemum mammoth, and the series would be mammoth. And then we might name each cultivar within that series a specific name, or it might just be the color name. So there might be mammoth red daisy or mammoth yellow daisy. So that series creates interest on the market. It's fairly small as far as its risk in the market and its economic reward. And so if we create new products that go within a series, they have, they have some economic value, but not extremely high in value. Then if you step to the right, you see a new series might come along. And so that might be from a competitor company or a public sector breeding program. And the new series that comes along is somehow enhanced from the current series that are on the market. So it might be earlier flowering time for a grower. It might flower all season long for the consumer, etc. It has something that's been identified that has market value. As you see, that moves up the scale as far as risk and economic reward for the breeding programs. And so there's greater chances of increased sales and royalties for the producers. Then above that is a distinguishable product, which is a kind of a step beyond what is normally found in any product class. So like it might be an impatiens that comes along, and suddenly we have a new form that moves the impatiens into a new arena, like maybe it's a new flower type. So those would be called distinguishable products. And then in the top right-hand corner that's circled in red are the ones that have the most economic reward or most value, but also represent the highest risk, which means that they're oftentimes long-term breeding objectives. They may be interspecific hybrids, and it creates a completely new habit for the product. And so that might be like our shrub chrysanthemums and mammoths. It might be like the wave petunias that changed upright petunias to being ground covers or wave types. Those types of things are of interest to our particular program since we're interested in long-term high-risk breeding. So the products that I'll be talking about today would fit within the category of being innovative products. Next. What are we looking for in addition to just creating something that's new or novel? So here are some of the traits that are of interest to gardens and gardeners. And you can see in some flower display trials of new cultivars a vast array of color and plant habit and shape and so forth. So cost, of course, is a driving factor first. Then novelty, something that's new, that's going to cause you to slam on the brakes when you drive by it in someone's garden and say, what is that? I must have it. So that kind of really eye-catching novelty. And of course, with annuals and particularly with perennials too, if we can get it, we'd like to have instant color. They can just plant them in the garden and have that instant look, as well as having them bloom throughout the entire season, preferably with low maintenance and other types of traits that might come along. Next. And of course, the current generation of consumers, the Gen Ys, are looking for new products as well, based on what they're expecting from the standpoint of products in horticulture and in particular flowers. So they're used to working with sound bites and info bites, and now we think in terms of flower bites for Gen Y, like this miniature chrysanthemum on the top right-hand side, and basically things that are fast, easy for them to work with in the garden, and that match some of the trends that they're certainly interested in, like being locavore or something that's sustainable, etc. And the idea of having lazy, efficient gardening products for them is certainly something of interest. And in the line of Gladiolus, for instance, we've created our first winter hardy Gladiolus, which fits into the category of lazy and efficient gardening, such that in the north, gardeners wouldn't have to dig the corms in the fall to preserve them over the winter. They could instead have them survive in the gardens. The bottom line for Gen Y, as well as all of the consumers, is two words: flower power and convenience. So we want lots of flowers, basically covering the plant as much as we can. If you see in the top right-hand corner, you see mainly flowers, and then, of course, convenience for everybody. Next. And so flower power and convenience drive what the private and public sector breeding programs are always focusing on for the U.S. and global consumers. Since gardening is now the #1 U.S. hobby, and everyone spends lots of money on ornamental products that enhance their garden and their lives. Next. Just some examples of what we work with if we have a specific crop or product out on the market. And this is an example of impatiens, the #1 bedding plant in North America. Just some examples of flower colors that can be bred by flower breeders and color patterns. So if you look in the bottom center, you notice like a star-shaped or stardust pattern. That was developed by Pan American Seed Company. Others, like the bottom left, have an eye in the center of them, and others just have nice pure colors. Next. And just to give you an idea, within the species of impatiens, or Impatiens walleriana, here's an example of some of the color types and patterns that are bred by flower breeders to create novelty and interest within the market. We try to strive to get every possible color palette in any product that is brought to the market to create continuing interest within a series or in a new series. Next. And here's just an example of what commercial seed production is like in the floriculture market. This is in Gilroy, California. This is an example of marigolds that would be growing as open pollinated seed for production to the market. And these are French marigolds, or Tagetes patula. Next. Just to give you a view of what the distribution chain within horticulture is like, and that includes both flowers and all other horticultural products. You start at the top with the plant explorers that go out into the wild and collect germplasm that may be of interest for the market, and then it moves into product development where the public and private sector breeding programs are. And that, of course, is what the perspective that I will be speaking from today. And then from there, the various types of products below that, the seed and vegetative products, moving down through the system with marketing firms and branding and brokers, through producers, distributors, commercial growers, retailers, all the way down to the consumer. Next. So what are we looking for? Well, as I mentioned, we've been an active breeding program for almost 100 years here at the University of Minnesota. We've been working on chrysanthemums for a long time, and now we're working on new types of products that will have interest for Gen Y and successive generations of consumers. Next. So the University of Minnesota flower breeding program is one of the oldest continuously operating public sector breeding programs. These are just some historic shots of trials of our products through the years, through 1930s onwards, and some of the persona involved with that have worked with chrysanthemums and various other products. Next. To date, we released 110 cultivars from the program since 1939, which mainly include chrysanthemums. And like I mentioned, we're moving on to new areas. So we have a new Gladiolus and Gora, for instance, that are on the market. Next. So the first crop we're going to focus on are lilies. These are interspecific lilies in the genus Lilium, and the interspecific name from is Lilium X formolongi, which are the crosses between Lilium formosanum, top left, and Lilium longiflorum, which is the Easter lily, which you can see in the center there in a pot. And these two white trumpet lilies are closely related. They're both from Japan. And we discovered this product back in 1999 as an interspecific hybrid on the market. And students in my potted plant production class grew them up because we wanted to see if they really would flower within the time frame that the companies were advertising for. And we noticed that indeed they did. They flowered in about 200 plus days from sowing. And the interesting thing was, though, in Japan, they're just used as cut flowers, harvested once and thrown away. And so we wanted to know what happened if you kept the plant growing. So we transplanted the seedlings from the greenhouse after they had flowered into the field, which you see on the top right-hand side, in the year 2000. And in between each orange stake is a single plant derived from a seed that spring. And you'll notice that there's multiple flowering stalks with multiple flowers on each plant. The interesting thing about the dates of this particular flower is that this is in a Minnesota field on October 31st of 2000. So this is after several frosts, and basically these have been flowering all summer long. As a result of that, we started a breeding program to work on continuous flowering of lilies that didn't require any cold treatment and that flowered in 200 days or so from sowing. And we've identified a couple genes that are involved in this flowering. And basically, you'll see on the bottom left-hand side, there's some seedlings in plug trays that are being germinated. Each of them have different colors of toothpicks. And we do selection in the plug phase for early germination. So each color of toothpicks means that those seedlings have come up in different weeks, so that when we come to transplant them, we only transplant the early germinators. with a specific color of toothpicks. And then in the middle, you see a picture of seedlings growing up after they've been selected in their plug trays. And then to the right, just an example population of seedlings that are flowering in about 180 days from sowing. And these will continuously flower actually for years if you keep them in the greenhouse. In the garden or in the field, they'll also flower all season long. We have identified two genes, VER1 and VER2, that are responsible for this phenomena of being able to flower without cold within the first year after sowing. And you might say, what's so interesting or unusual about that? Most lilies, when they are crossed and then sown from seed, require 3 to 5 years to flower. And so this is a phenomenal hybrid in that it can flower in less than a year from sowing. Next. So just to give an example of the action of the VER1 and VER2 genes, on the left you have a population, a set of hybrids that do not have the dominant alleles for those two genes, or those two loci, which means they require cold in order to flower. And these are the same age as the flat on the right, and those on the right have dominant alleles at VER1 and VER2, and being exactly the same age, however, they are tall and in flower. So the ones on the left, if they don't have the dominant alleles, require a cold vernalization treatment in order to elongate and flower. Next. Our early objectives for the first 10 years in working on these were to try to get color introduced into these hybrids. Because as you notice, they're all white trumpet lilies. So we looked at a variety of different techniques to get that from other species that would cross with these Lilium X formilongi. And basically the one that worked the best for us of the three options that are listed there are working with Lilium martagon. There's a picture of a Lilium martagon, or a Turk's cap lily, on the right with the red recurved petals. And that's because this particular species actually in the wild or in the field or in your garden will initiate flowers in the fall prior to winter or cold, even though it's not very closely related to our species. So through a series of complex interspecific crosses and so forth, we were able to incorporate Lilium martagon into our hybrids and start to achieve some color. Next. So the process that we use to do this is pretty complicated. So we did crosses with those parents at the top bullet to create a very complicated hybrid that's in the brackets there, 00L111-343 times a different parent. Then we crossed it with a variety of different martagon parents that had good general combining ability, or GCA, which meant that they crossed really well with a wide variety of parents. We crossed them in both directions, whether it's the martagon is male or the martagon is female, with our Lilium X formilongi. We used a cut style technique that's developed, and you can see that in that small picture to the right of that bullet there, the orange flower bud that's been cut off, and the style is there but partly cut off as well. So we apply the pollen to the cut end of the style, and there's a very short space to grow until it reaches the ovule and can affect fertilization. And then we use embryo rescue, or ER, which is basically taking the ovules out of the ovary after fertilization, a few weeks after fertilization, and getting them into tissue culture. You see some of them there on the right, lower right-hand corner there, working on at a hood under a microscope, doing embryo rescue and getting them into culture. And then we basically grew up a variety of sets of hybrids to see and screen them to see if they had color or not, as well as flowering without cold. Next. So the most successful interspecific cross was this complicated pedigree that you see on this particular slide. And this was the hybrid that we just termed O7L, which meant we made that cross in 2007. And L just stood for Lily, and that was cross number 14. So that's the complicated pedigree that you can see. And basically at the bottom, we end up with this new hybrid, O7L-14. We had the F1 hybrids, and then we also created some F2 hybrids. And we had 111 plants or seedlings that arose from this particular cross. So we grew those up to see if they required cold or not. In other words, if they had dominant alleles for Ver1 and Ver2, as well as did they possess color in their flowers. Next. We also did molecular screening of these to determine where these all fit in relationship to the parents. So this is an example dendrogram of intersimple sequence repeats, or ISSRs, that are looking at small sections of the nuclear DNA within lilies to determine the differences. So you can see the female parent is circled there at the top, that 00L111-343. The male parents, the Martagon parents, are right there in the center, highlighted in green. And then all the other plants that go O7L-14 with an additional number are the F1 hybrids that we screened. Most of the hybrids flowered, so they had dominant Ver1 and Ver2 alleles, but they had white trumpets. They were not colored. However, the bottom set down there that we are terming recombinants did not flower that first year. So then we put them into cold or vernalization and then grew them out. And lo and behold, all those particular plants had color in the petals. So we were successful in getting color. However, they didn't have the dominant alleles for Ver1 and Ver2. Next slide. And so in the upper right-hand corner is just an example of one of the lilies that had color, a colored throat, and it wasn't a trumpet lily. And that was one example of some of the recombinants. And then the next picture halfway down on the right-hand side there, you can see that a lot of them also had this very long thread-like leaves that were very atypical for lilies. So what we're working on now, and this is a long-term project, is we're going to be sequencing the Ver1 and Ver2 genes and then characterizing these recombinants to see what alleles they're missing and see if we can incorporate dominant Ver1 and Ver2 alleles into the recombinants so that they flower without needing cold, and then continuously backcross them and select for enhanced flower color such that we can get pure colors throughout the entire petals of these particular recombinants. We're also going to try to determine heritability, or H squared, of all the various traits that are unique within these recombinants, and then assay them for a variety of known mutants in tobacco or Nicotiana and another grass, Oryza, to see how they compare with established mutants. So that's kind of where we're at in the development of these lilies. Our long-term goal of having seed-propagated colored lilies that flower the first year without cold is making progress. It's just a few years yet before we actually have that product. Next. Second, I want to talk about Gladiolus. And as I mentioned, that we now have winter hardy Gladiolus on the market. Our first cultivar is Beatrice. It's on the lower left-hand side there. That's a 3 to 5 year old corm that's flowering, and it will survive in Zone 4, USDA Zone 4 or higher, or USDA Zone 3 if it's properly mulched and there's adequate snow cover. And so right now we're working with potential licensees to establish North American production chain for this corm or geophyte to bring it to the market. Next slide, which actually will be a pop-up of another Gladiolus shot. And you can hit next as well. And that's actually a year 3 plant. So on the left, you have a plant that's, the corm is 3 to 5 years old. That's the first year in the field. And then on the right, here's what it looks like after 3 years in zone 4. And you notice there's multiple shoots coming up, multiple flowering stalks, and it basically fills in with just lots and lots of gladiolus. Next. And how do we determine winter hardiness? It's a long process. We can trial them in the fields, provided we have a cold winter and adequate snow cover. But with global warming, that's quite variable. So instead, we do programmed freezing tests in the laboratory. And we basically freeze them down to our test temperatures for USDA Zone 4 or Zone 3, which, if you look in the table there, would be minus 10 C is the test temperature for soils for plants to survive in USDA Zone 4, and minus 12 C is the test temperature for survival in USDA Zone 3. And basically, we'd want fairly high scores for the parts of the corm, tissues, and so forth. These are on a 1 to 5 scale, so we'd expect 3.5 or higher to have adequate survival. And you notice that we do have numbers within those ranges for those 2 sets of critical test temperatures. Next. And this is just an example of some smaller sized corms that are frozen to varying temperatures from 0 C to minus 12 C. And you can see the relative amount of damage of some and not in others. Like on the bottom row, like for instance at minus 12 C, you can see a very healthy corm that's still alive after freezing to minus 12 C. Next slide. We wanted to know how related this winter hardy one species was. This cultivar Beatrice was to all the other gladiolus that we had in our breeding program. And if you look in the very middle on the right-hand side, and again, this is an ISSR dendrogram from nuclear DNA. And if you look at VT03, which is Gladiolus Beatrice, it's very closely linked to the one right below it, which is GL-DAHL, which stands for Gladiolus dahllenii, which is a fairly winter hardy species from the historic records. And then all the other species and accessions that are in there are less related to Beatrice. So we're using Gladiolus dahllenii as an additional species to create related winter hardy forms of Gladiolus. Next slide. And we've been determining the heritability of winter hardiness. And basically H squared values range from -1 to +1. And the closer they get to +1, the more heritable it is. And you can see that for the traits that we have here for the number of roots, root length, corm damage, and shoot length, that most of these traits are not very heritable in Gladiolus, which means our work is going to be very difficult. And so as a result, it's going to take us longer to create new winter hardy cultivars of Gladiolus. So that's one of our long-term objectives. Next. So what we're looking for is winter survival and a variety of other traits in Gladiolus. So let me show you what we've discovered in this process. Next. One of the things we've discovered is that corms may be winter hardy, but they may not have good performance. So on this slide on the left, you see we have a few Gladiolus. You see those long strap-shaped leaves that came up, survived the winter, but never flowered, versus one on the right-hand side that not only came up and survived, but also had multiple flowering stalks. So sometimes surviving through the winter gives you green shoots, but no flowers. And of course, what we really want to select for is what's on the right-hand side. So that's one example of a winter hardy form that we really want to have. Flowers, of course. Next. So we always ask the question when we're making wide crosses, is why not get some wild color combinations, or maybe we can find something new that's never been seen on the market before. So these are two examples of new types of gladiolus flower colors or petal types and so forth that we have amongst some of our interspecific hybrids. Next. Here's some additional types, and these actually are hybrids with the species Gladiolus dalenii, which has a characteristic sort of hood petal at the top. And if you look at the pink flower on the right, you can see one of those petals has a nice hood covering the center of the flower. So that comes from the species Gladiolus dalenii. Next. We were also trying to do other things like perhaps decrease the space between the leaves or the flowers within the flower scape, which is basically decreasing internode lengths, as well as adding what we call flower surround. Typically, gladiolus are what we call faced. In other words, the flowers all face in one direction. And we were thinking, wouldn't it be fun to get some gladiolus flowers that actually surrounded the stem, faced in all directions? And so here's one example on the right, this kind of reddish-orange streaked hybrid that has flowers, as you can see, that are going around the stalk, versus one in the background there on the right that's faced. They're all facing in one direction. So this was one of the things that we were selecting for, and inadvertently, as oftentimes happens in plant breeding and in particular flower breeding, we discovered something new that we'd never even dreamed of. Next slide. And that was dwarf gladiolus, where we had not only decreased the internode length in the flower stalk, so that the flowers were all tight together, but we also created a dwarf plant. But the interesting thing is we have very large flowers. So if you look at the one on the bottom left there, the pink with kind of a yellow throat, the total height for this plant is 22 centimeters or 9 inches. And the one on the right was a bit taller. That was about 11 inches, the red one. And you notice we've got two different types here. So the one on the left that's 22 centimeters tall has flower surround. So the flowers actually go around the stem. And the small picture to the right shows several examples of those, of this particular hybrid. Whereas the one on the top right-hand side, the red with the yellow throat, is faced. So the flowers are only facing in one direction. So as we looked at these in the field, we were thinking, wow, this is a completely new product. There are miniature gladiolus on the market, but they look nothing like this. And in fact, we thought these would actually be very interesting to create a completely new product for the market, these dwarf flowering potted types. So you could actually buy flowering potted gladiolus in pots on the market, like you do a lily or a chrysanthemum. Next. And so what we've been doing is a series of fast, rapid generation cycling to speed up the breeding process for gladiolus, because we don't have VER1 and VER2 genes in it like we do in lily. So it takes 3 to 5 years from sowing to flowering in order to create a flowering hybrid from seed. So we've had these selection strategies that we've developed in order to be able to do this. And if you look at the bottom, you notice that we basically, in our whole process, which I won't step through, we've been able to generate as many as 3 generations per year with our truncated breeding and selection process. Next. And so our gladiolus breeding objectives for the next few years have to do with, what's the heritability of these compressed stems and our dwarf potted types, as well as whether or not there's linkage with winter hardiness and other sorts of aspects of potted plant gladiolus that are of interest. So our goal in the next few years is to create a series of dwarf flowering gladiolus that would be appropriate for potted plant production for the market. Next. And with that, I will stop and entertain questions. I just wanted to thank all of our sponsors for our many years' worth of work here at the University of Minnesota. Thank you for joining us today.
Well, thank you, Neil. Now we'll have quite some time for some questions. For those of you who missed the beginning of the presentation, you can use the question box on your screen to type in questions and then hit return. If the question box is closed, you can click the small plus sign next to the word question to open it up. We'll be reading the questions out loud, and then we'll answer as many as we have time for. So we already have some questions posted, and I'll go ahead and read those to you. Is there an advantage to hybrids, or is there an advantage to F1 hybrids, or is this a way to protect varieties? And that was a question from David Francis.
Well, in answer to your question, David, the answer is yes, in both cases. So the pluses of F1 hybrids are that they combine the good traits of both parents and really enhance it and have much more vigorous growth and performance as an F1 hybrid. And it's also a method of protecting the plants. Basically, any private or public sector breeding program that creates a new F1 hybrid, particularly if it's from seed, they own the parents for the hybrid, and so that offers them protection. So nobody else can create that exact F1 hybrid because they own the parents.
All right, thank you, Neil. And then the next question was, what's a good text for finding information about breeding various species of flowers, if there is one, and also what are some good resources for germplasm? And that was from Kay Mueller.
Hi, Kay. In answer to your question, there's a couple books that are out on the market that have to do with how you breed and develop flowers. So one of them I've actually edited myself, and the title of that book is Flower Breeding and Genetics. And that's published by Springer in the Netherlands, and that's available on Amazon.com. So that's Flower Breeding and Genetics, and I'm the author on that. There's also another book that's called Breeding Ornamental Plants, which was written by Dorothy and M. Brett Callaway. That's Callaway. And another book that might be of interest would be a book called Double Flowers. That's by Joan Reynolds and John Tampion, Tampion. And as far as germplasm information, probably the best sources of germplasm that's available within the United States would be what the USDA, or the U.S. Department of Agriculture, has available on their Germplasm Resources Information Network, or GRIN. So you can just look up USDA GRIN online and get right into their system. They curate various types of flowers, but not all flowers. And then other germplasm networks would be basically, I mean, what we do, sometimes we collect materials ourselves. You can get them from collectors. There's lots of amateur societies that specialize in a genus, like there's a Gladiolus society and a Lilies society and a Chrysanthemum society. So those people oftentimes have great resources for finding plants that might be of interest. And then there's also various societies that have seed exchanges, like the North American Rock Garden Society every year has a huge seed exchange list. And so sometimes you can find rare and valuable types of ornamental plants through that. And you can just search for that society online.
All right, thank you. And a question from Dan Parfit. How does the University of Minnesota handle patenting and royalty distribution, et cetera?
Good question for a public sector breeding program. We've been patenting vegetatively propagated cultivars since about the 1970s here at the university. And basically what we do is if we have a new cultivar or hybrid that comes onto the market, and it's propagated via cuttings or asexually, such as all chrysanthemums would fall into that category, and our gladiolus would as well. We will file for a U.S. plant patent on it, and then we will license it to licensees or a licensee if it's exclusive, and they will be responsible for propagating it and getting it into the whole horticultural distribution chain.
Okay, and from Keith Williams, understanding general combining ability was mentioned as a part of the lily breeding program. So how many testers are you using, and or how are you assessing general combining ability in your new lines?
Hi, Keith. Good question about general combining ability. We have a variety of standard testers that we use, like in our lily breeding program. So we have about 15 to 20 testers that we use to determine the ability of a particular parent to cross and produce seed with these testers.
Okay, and from Mohan Narula, I hope I'm pronouncing that right, do lily and gladiolus have male sterility and double flower traits? If yes, how are they inherited, and have there been molecular markers developed for them?
Hi, Mohan. Okay, so that's a series of questions. So are there double flowers present in lilies and gladiolus? In lilies, that's a fairly recent phenomena, and typically it's anthers get converted into petals, and so you can have double flowered lilies that pretty much just have petals, but no pollen or anthers present, and sometimes they don't even have a style present. So some of those actually are sterile. There isn't any defined male sterility system or female sterility system identified in either lilies or gladiolus to date. So we can't do the same sorts of things like we could with onions or something using sterility to create F1 hybrids from seed as of yet. In gladiolus, we have, I guess we'd call them at best semi-doubles, where some of the anther tissue is converted into extra petals. They're not complete doubles, I wouldn't say. They're quite variable in the number of petals that they have. So we'd call them kind of semi-doubles, I guess would be the best way to term it. And as far as sterility in gladiolus, I'm not aware of any true sterile gladiolus amongst any of the cultivars or parents that might be available. And nothing has been sequenced to speak of in either genera, so that's kind of a whole new venue that we'll be exploring in the breeding programs.
Okay, thank you. And from Emily Clarquist, you mentioned that the heritability ranged from -1 to +1. How did you obtain negative heritability values given that it is H squared?
The negative values come from the range of heritability values that come out, and so yes, I mean typically when it's squared, it should all be positive, but in some cases we can get numbers below 0.
All right, and I have a question from Kim, and my question goes back to the slide at the beginning of your talk where you showed how the number of cultivars in the market has increased dramatically over time. And
Right.
I was wondering, is there any data out there on what percentage of those cultivars are ornamental?
There isn't actually any data on that. We never, in the paper that we published on that, we didn't have the ability or the manpower to go through and actually determine that. But it's a significant portion, I would say greater than 50% of the cultivars that are on the market are ornamental. But we don't have the exact number. So it's a
All right.
lot, in other words.
Yeah. And then my other question related to that was, do you know how many cultivars, ornamental cultivars, are the product of a breeding program versus just random chance seedlings?
That's a good question. There are a lot of random or chance seedlings or mutations that arise, and oftentimes breeding programs will maybe develop a new hybrid to create a series, and then as a fast way of getting additional colors to match the series, they'll try to create mutants. So they may actually submit them to ionizing radiation, or they may put them through callus culture and tissue culture, and that will be enough genomic shock to create new colored hybrids. And I don't know that anyone actually has the actual estimates of number of mutants versus actual seedlings that have been bred directly on the market. I'd really be hard pressed to guess if it's an equal split between mutants and non-mutants or not. I really couldn't even tell you. It's going to vary by product, and whether it's seed or vegetatively propagated predominantly. So if I had to venture, at least within the realm of vegetatively propagated crops, it would be a fairly high percentage that are mutants. But I couldn't give you a number.
Okay, thank you. And if I guess no one has any more questions, we can go ahead and wrap up the webinar. So thank you so much, everybody, for joining us. And Neil, thank you for sharing with us. And everyone have an excellent day, everyone.